guys, so we're going to go over our notes for meiosis. So this is dealing with cell division. So let's review, why is cell division beneficial to an organism? Okay, so we need to be able to repair damaged or dead cells, to grow because we need to make more and more cells as we go throughout our lives. And one thing we didn't really mention before in our previous unit was asexual reproduction. So some cells reproduce by cells dividing, and that is kind of like making a clone or just asexual reproduction. So mitosis, which we talked about last unit, makes identical cells, and this is because we don't want our skin cells having different DNA than other skin cells. We want them to be able to go through and be produced as identical cells with identical DNA, and that's why we have those mitosis checkpoints. So here's just showing a cell go through mitosis. Okay, so advantages of asexual reproduction versus sexual reproduction. Well, we're first going to go through asexual reproduction. So asexual reproduction can happen fast. It can happen with very little energy required. And there's many different types of asexual reproduction, and this helps especially if there's a lack of mates, and we don't have to spend as much time finding mates. The disadvantages, the big thing is there's no genetic variety. Okay, so this is bad for our evolution and natural selection, which we'll talk about later in the year. But if we have a lot of organisms that are identical, something could happen and just wipe them all out very easily. And the clones then that would be produced are going to compete for resources. And this would be specifically for stationary organisms that don't move around. And then adaptation and evolution would be slowed down, which I mentioned, was talking about our natural selection. So now we're going to shift gears in this unit and talk about sexual reproduction. And so how that happens is we have to have cells that are haploid, meaning they have half the number of chromosomes, to be able to combine together to make an offspring. Okay, so here we have showing our egg and sperm that come together to create a zygote. This is sexual reproduction. So we're going to focus on how humans make these cells. So first of all, we're dealing with genes. So genes are those segments of DNA that code for the basic heredity. So offsprings require genes from parents by inheriting those chromosomes. So eukaryotic DNA, like humans, is divided into numerous chromosomes. Humans, for example, have 46 chromosomes, which you will need to know. Dogs and tigers, you don't need to know those, but they have a different number, 78 or 38 for a tiger. And then this fern here has 1,262. So depending on the organism, depends on the number of chromosomes, and it doesn't necessarily mean that a more complex organism has more chromosomes. The number of chromosomes has really no bearing on the complexity. So we can see we have pea plant here that has only 14, but a dog has even more. Sunflower is down here, pufferfish, human, 46. Okay, so we can kind of map out our chromosomes in what is called a karyotype, and you've probably seen these before. This is where our chromosomes are lined up. So here are human chromosomes. So how many human chromosomes are there? Okay, there are actually 46. You might notice that each one of these are paired up, and so it might look like there's 23, but there's 23 pairs or 46 total chromosomes. So half of the information humans receive is from their mother and half is from their father, which is why we have 2 copies of the same chromosome. There are just different variations. So we have 1 copy from mom, 1 copy from dad for chromosome 1. Same for chromosome 2, 1 copy from mom, 1 copy from dad. These are called homologous chromosomes because they have similar information on them. So maybe chromosome 1 has gene information for hair color and eye color, but chromosome 8 has information on height. And that's just really an oversimplification, but it's got the same type of instructions. It just might have different versions of those instructions. So how many pairs of homologous chromosomes do humans have? We would have 23 pairs of chromosomes. Karyotypes are organized with homologous chromosomes together, and they're also organized from largest to smallest, as we see here. So in humans, 1 through 22 are called autosome or autosomal chromosomes, and the last one, the last pair of homologous chromosomes are sex chromosomes. Okay, so a couple other terms we need to know that you might remember from regular biology. Somatic cells, these are our body cells. They have 2 copies of each chromosome, and we refer to them as being diploid. Diploid is just referring to that there are 2 chromosomes for each homologous pair. They're represented as a 2N for that diploid number. So in this picture, we have 4 chromosomes. So, and there's 2 different versions, but you can see there's 2 large and 2 small. So in that case, we have 2N or we have 4 chromosomes. So it's a diploid. The homologous chromosomes, again, have the same genes, but they might have a different version or variation. These are going to be where our alleles come from. So alleles are different versions of the same gene. So maybe there is a gene for seed color, and there's an allele for green and an allele for yellow. This is an example of how we can have 2 differences on 2 different homologous chromosomes. So here's an example where we would have this A, B. This is for blood type. So someone could have AB blood if they have 1 allele A and 1 allele B on those 2 different homologous chromosomes. Each chromosome has specific genes on them. They're going to have multiple genes, and every person has the same exact genes in that exact same spot. So maybe this is chromosome 1, and there's a specific location on the chromosome that is for this blood type gene. It would be on that same location on chromosome 1 for every individual. Here's kind of an example of what this would look like, where we have different bands representing our different genes. And here it actually is chromosome 9, is where that ABO blood group is. So genetic genes are important because they're the genetic information on how to produce proteins. So this gene will then be used to create proteins, and that will determine the blood type. So what about sex cells? They don't have the same number of chromosomes in our body, because if they did, then we would have 46 chromosomes in an egg, 46 chromosomes in a sperm, and then when they came together, it would have 92 chromosomes, which is no longer human. So because of that, it's very important that we half the number of chromosomes. So we need to go from 46 chromosomes to 23 chromosomes in those eggs and sperm cells. So this is because of this, we need to create cells or sex cells called gametes or gametic cells. These are the cells that are going to be used for reproduction, and something that's important about these cells is that they are haploid. Haploid is talking about it has half the amount of genetic information, meaning it only has 1 copy of each chromosome or half the number in a body cell. Okay, and they're going to be denoted by an N instead of the 2N for diploid. So to create cells that have 1 copy of each chromosome, a cell must divide twice. And we're going to go through this. This is the process of meiosis or meiosis. Mitosis is going to make those identical body cells that we talked about last unit. Meiosis makes non-identical cells, and they're sex cells or gametes. This is kind of vocabulary that we should be able to know, and it's organized into mitosis and meiosis. So mitosis makes body cells or somatic cells. They're diploid. They have 2 copies of each chromosome, 1 from mom, 1 from dad. Mitosis is also used in just asexual reproduction. Meiosis, on the other hand, produces sex cells, which are haploid. They have half the genetic information, only 1 chromosome from each parent, and then they are in gametes. They're creating gametes, and they're used in sexual reproduction. Okay, so we're going to go through the steps of meiosis. They're very similar to mitosis, so we're not actually going to go through every single step, but it's broken down into 2 phases, meiosis 1 and meiosis 2. So the big thing to notice is what is being separated. So here we have meiosis 1. See, we have 1 cell that goes and it splits into 2, and then in meiosis 2, we split those 2 cells further into 4 cells themselves. So the big parts that we're going to pay attention to are prophase 1, metaphase 1, anaphase 1, and anaphase 2, because this is where we can have some extra things going on. But interphase is just going to be like normal for mitosis, and then we'll go into prophase, and each of these is 1 for meiosis 1, and then we'll go through PMAT for meiosis 2. Okay, so the steps of meiosis. First, we're going to go through interphase, which I mentioned, still growth, DNA synthesis, and growth is again. Okay, so it's still going to be the DNA is duplicating, chromosomes are going to be connected together at that centromere. And then they're also called sister chromatids when they're attached here. The cells grow bigger. And so again, we have our homologous chromosomes here. They've just now replicated. So we still refer to these as sister chromatids. Remember that a sister chromatid is not the same thing as homologous chromosomes. Notice that here's one chromosome, maybe it came from your mom, and you have 2 copies now because we went through DNA synthesis. Each of these copies is a sister chromatid. Same thing happened with the other chromosome, but this maybe contains information from dad. So we now have both homologous chromosomes duplicated. So the homologous chromosomes, there's one version of chromosome 1 from dad and one version from mom. And our sister chromatids, yeah, that's the copies. Okay, so I'm actually going to switch back over here. Okay, so you can see we're going to do our paper plate model. So we have, at this point in interphase, our DNA has not condensed yet. So we are starting out with 2 white strings and 2 blue strings. This is the beginning of our interphase. We have it in a nucleus. And then what's happening in this stage in interphase is that we're duplicating that DNA. So we'll add 2 more strands of blue yarn and 2 more strands of white yarn. So at this point, if we look at the cell, we cannot visually see the different chromosomes, but we have a replicated amount of DNA in it, and then the cell itself will start getting larger. Okay, let's go back. Okay, prophase 1, this is similar to our regular prophase in that the nuclear membrane is going to break down. The DNA will coil up into those chromosomes. A couple other things that are important that happen here that are not the same as in mitosis is the homologous chromosomes are actually going to bind together and create something called tetrads. And this process is called synapsis, where these homologous chromosomes are going to bind together. This is important to make sure that we have the homologous chromosomes in the right place so we don't end up with 2 chromosomes that have I information going into one cell and no I information going into another cell. So it's just helping us get the information in the right spot. So here is showing the homologous chromosomes lining up together. So DNA is replicated already. So blue is a copy from dad and red is a copy from mom. So you can see 1 copy from mom, 1 copy from dad are being lined up together. This is forming a tetrad. And the big thing that happens here too is something called crossing over. So these chromosomes are going to actually exchange information because you don't end up actually receiving 1 full chromosome from your dad and that happened to be all of this information from his father. You receive a mixture of information from your father's parents. So chromosome 1 is going to exchange information with the other chromosome 1. And this is also called homologous recombination because those homologous chromosomes are recombining. Okay, so here we're seeing the tetrad, the site of crossing over taking place. We still have those spindle fibers and everything happening from before, like normal in prophase, and our 2 sister chromatids for each one. So let me go back so I can show you my paper plate model. Okay, so we have condensed our chromosomes. So now we have our chromosomes being represented by pipe cleaners. They've condensed down. So remember, we have 2 sister chromatids and they're connected at our centromere. Here I'm representing two homologous chromosomes with two different shades of pink. So these are going to have similar information, just one came from mom, one came from dad. And so they're going to combine together and form a tetrad. And this process is called synapsis. And this is where it's going to exchange information. You can kind of think about how maybe this dark pink will come over here to the light pink, and the light pink will come over here to the dark pink. And I'm not going to actually physically cut my pipe cleaners, but you can imagine how we can have crossing over happen in different places and exchanging that information. So this would happen with the other homologous chromosomes. So maybe we had blue and purple here representing two other homologous pairs of chromosomes. Maybe they have information on eye color. And they're going to swap information. So we have a mixture of what's produced. Okay, the importance of this is also it's increasing variation. So we're having new combinations, which is helpful for us. Okay, metaphase 1. This is very similar to metaphase in mitosis. It's still metaphase for middle. Here our homologous chromosomes are lining up on the middle, or our metaphase plate. Those spindle fibers still connect to those kinetochores at the centromere. The big thing here is notice that the homologous chromosomes are lining up, not each individual chromosome. So this is important. They also line up randomly. So maybe this red one's over here and this blue one's over here, but they could have swapped depending on how they oriented. This is called independent assortment, the idea that they independently line up on that metaphase plate. Just because red is up here doesn't mean that red has to be up here for the other one. They each independently assort. So we have more probability and more possibilities for our combinations. So because of this, humans have 23 pairs of chromosomes. There's actually like over 8 million different ways that they can just line up. So that gives us even more variation in our chromosomes. So here I can show you we have our tetrads that are being formed, and they're going to line up on our plate, on the metaphase plate in the middle. So that this time when they separate, the homologous chromosomes will separate, as opposed to in mitosis where the sister chromatids separate. So that's how this is different than mitosis. And again, this is increasing variation. Anaphase 1, just like before, chromosomes are going to move and separate. Spindle fibers are going to pull them. This is where our tetrads separate in this case, and not our sister chromatids. So the sister chromatids are still connected at that centromere. So crossing over happened during that prophase 1. So when they separate, they've already done crossing over, and all those sections that they exchange information with are already shown, which you can see here why there's this little bit of blue on this red chromosome and this little bit of red on this blue chromosome, where we had that exchange of information in crossing over. It's mostly from the paternal, but it has a piece of the maternal swapped in. That's a way of thinking about what's happening in crossing over. Okay, next is telophase or telophase 1. So it's going to be very similar to what happens in regular telophase, where we're going to be terminating what's happening. So we're going to have cytokinesis occurring. Afterwards, we're having our 2 sets of chromosomes. We now have 2 cells, though, however, that are not identical, because we had that crossing over happen and we had that independent assortment. Okay, but we're not done yet, because our goal of meiosis was to have cells that were half the chromosome number, and we actually did half the number of chromosomes, but we only want 1 copy. We don't want extra copies. If we have extra copies, we can have errors in that. So what can we do? We're going to do the process again. Okay, so the process of meiosis is going to go through that division twice. So now it's already half the number of chromosomes. It's like Groundhog Day. We're going to do it all over again. Okay, so Cells are going to skip interphase this time though. And why is that? Think about that for a second. Interphase is where the DNA is synthesized again. If we were to synthesize more DNA, we would never get out of this loop of having to split our cell more and more to get half the amount of DNA. So we don't want that extra copy of DNA. But we do need to have it happen for that first time. And the reason we have that interface happening that first time is we need that homologous recombination and that independent assortment happening in meiosis 1. So that variety is happening in meiosis 1. Because we want to have a nice new mix of DNA for each offspring and for each cell. Okay, so now we're going to continue on. We have 2 cells and they both have 2 sister chromatids, and they're crossed. We had crossing over happen, so they're all mixed up. And they're haploids. They only have half the number of chromosomes, but we have 2 copies. So now it's just going to be just like in mitosis, but we won't have crossing over, and it's just going to split. So meiosis 2 is basically mitosis without interface. We're going to skip that synthesis stage. They're going to line up in the middle. They're going to separate, and we'll end up after telophase and cytokinesis with 4 different cells, all haploid, all with only 1 copy of the information. So now this time we're separating sister chromatids and not homologous chromosomes. The tetrads or homologous chromosomes were separated in meiosis 1. If you see homologous chromosomes, always think about meiosis 1 because they've already been separated by the time we get to meiosis 2. And again, remember in telophase 2, we're having that nuclear membrane forming back around the cells. Okay, we have gone over meiosis, and it occurs in the end result. But what is the end result? That's our sperm and egg cells. So it doesn't actually happen perfectly like this. For sperm cells, we end up with 4 sperm cells. And males who've reached puberty will produce sperm cells their entire lives. The older the male is, the higher the chance that the sperm may have more mutations in it, because there's an average for about 1 mutation for every 8 months. And environmental factors can affect the number of mutations, like tobacco or temperature, and alcohol can affect those sperm mutations as well as sperm count. Eggs are, on the other hand, are a little different. They actually don't quite divide evenly. So instead of ending up with 4 cells, we end up with 1 large cell that will be developed into the egg. And the other ones are called polar bodies, and they're kind of non-viable cells. So a female is born with all the eggs that she will ever have, roughly 2 billion. And by puberty, we have about 80,000 that have survived. But these eggs have not finished meiosis. They are kept at prophase 1 until ovulation, and then they can complete the rest of meiosis. So mutations increase with age as well. The older the individual is, the more likely that they'll have an error or a mutation, as well as an error in separating those chromosomes out. And that's how we can end up with too many or too few chromosomes, which is actually what causes Down syndrome. Again, cigarettes, lead, radiation, all of those can affect the rate of mutations in meiosis. So kind of a big picture of the life cycle. We have mitosis occurring. Then we can have individuals who have grown, and then they will go through meiosis, have half the number of chromosomes. Then they go through fertilization, where we're going back to the same number, and then we can do that all over again. So fertilization and meiosis alternate in sexual life cycles. So it will go meiosis, and then fertilization has to happen to get us back to that same number of chromosomes. So meiosis halves the number of chromosomes. Fertilization combines them so that we're back to our 2N, forming a zygote. So zygote divides by mitosis, then to continue the organism's lifespan. So we're going from egg and sperm to create a zygote, which is diploid, and that zygote will grow and grow and grow and create an offspring. Fertilization is also random. So this is another way we have increased variation because we don't have any control over which sperm and which egg are combined. So here we have mitosis and meiosis and how they are different. So we both have our parent cell, and we have chromosomes being replicated in prophase and in prophase 1. In mitosis, we have 2 sister chromatids for each one. In meiosis, in prophase 1, we have that tetrad forming in our synapses of homologous chromosomes. In metaphase, we have chromosomes for mitosis being lined up, and we have our tetrads being lined up in meiosis. In anaphase and telophase are very similar, but we have our sister chromatids separating in mitosis. We have our homologous chromosomes separating in anaphase 1, and then sister chromatids remaining together until our meiosis 2 when those sister chromatids separate. Our daughter cells of mitosis are referred to as daughter cells, and these are still also daughter cells of meiosis 2. It's the same type of term. Okay, so that is basically it. I hope this is helpful. Bye, guys.